This method provides a high-throughput platform for the screening of Salmonella and Shigella.
The main advantage of this technique is rapid, specific, sensitive, and high throughput.
This technique combines an AAT-based method and culture, in which real-time PCR screening was applied first, and then positive ones were sent for bacterial culture and identification.
Though this method focuses on Salmonella and Shigella screening, it can also be applied to other pathogens, such as Diarrhea, Staphylococcus, E. coli, and etc.
Visual demonstration of this method is critical, as the selection of the correct colony is difficult due to background flora in samples.
To begin, add 3 milliliters of nutrient broth to each sample. Then incubate the samples at 36 degrees Celsius for 6 hours. After this, centrifuge the pre-enrichment culture at 800 Gs for 2 minutes. Then, transfer the supernatant to a new tube, and centrifuge it again for 5 minutes at 12,000 Gs. After discarding the supernatant, add 100 microliters of DNA extraction solution to the pellet. Then vortex the tube for 1 minute. Next, heat the sample at 1000 degrees Celsius in a dry bath. After repeating the centrifugation, collect the supernatant into a new tube. First, create the reaction mixture and set the cycling program according to the text protocol.